Speaking about the basic knowledge in Wiskott, I believe that really the guy who should speak is with us here. It's Professor Adrian Thrasher, and just to give a very short bio about Adrian. Adrian is a professor of pediatric immunology at the University College of London, Institute of Child Health at Great Ormond Street Hospital, which we all know and appreciate. He finished his medical school at St. George Hospital and then did his PhD at the University College of London. He is one of the leaders in the field of PID, and his main interest is in gene therapy in several PID conditions, including WASP. He is also very active in studying the pathophysiology of Wiskott-Aldrich syndrome and has published numerous papers in the field. So, Adrian, I invite you to give us a talk, as you called it. First, the question mark should be deleted.
Well, maybe not.
I hope so.
Thanks very much, Amos, for that kind introduction, and thanks very much, Amir, for inviting me to this excellent meeting. So I guess my task is really to set the scene, and what I want to do is to try and explain to you a little bit about the biology of Wiskott-Aldrich syndrome, what we understand about the defective gene and the defective protein, and how that translates into human disease. And that's important because unless we do understand the background pathophysiology, it becomes much more difficult to design effective and novel therapies. Now the question mark, actually, I just forgot to delete it. But I'll see how I go. So I'm going to cover a little bit of ground, and if I don't get to the clinic bit at the end, we can tack that onto my last talk, which will be much more clinical. So what's deficient? This is a rare disorder. And many of you will have seen one or two cases maybe. It's characterized by these main features: microthrombocytopenia, which is invariable, and all that means is the platelet count is low and the platelets are small. Now there's no other disease that I know about where the platelets are small. So this is an easy diagnosis to make just by looking down the microscope, and it's amazing how many patients still arrive in my clinic with a diagnosis of ITP, and it's clear that they have Wiskott-Aldrich syndrome just by looking down the microscope. These patients also have complex immunodeficiency, which I'll talk about a little bit more. They may or may not have eczema. It's variable between patients, and typically they develop autoimmune phenomena, so these may be autoimmune cytopenias, hemolytic anemia, autoimmune thrombocytopenia on top of the intrinsic thrombocytopenia, but also other autoimmune phenomena that tend to develop later on in life, such as nephropathy and colitis. And also later on in life, these patients are more susceptible than normal individuals to lymphoreticular malignancy. So the thing that really kills these patients, if they're not treated by some form of definitive therapy, are the malignancy, severe autoimmunity, and severe infections. And that's in the classical form of this disease. Now, originally there were two conditions, X-linked thrombocytopenia and Wiskott-Aldrich syndrome. And when the gene for Wiskott-Aldrich syndrome was cloned back in the 90s, it became apparent that these are in fact variants of the same condition. So X-linked thrombocytopenia is a much milder condition. These patients have thrombocytopenia, but very few of the other features. So they tend not to have immunodeficiency, and they tend not, to a great extent, to develop malignancy or autoimmunity. And I'll talk about that in a lot more detail in my second talk. And we can score these patients on a scale from 1 to 5, which is of some utility, but not of great utility, I don't think, because patients actually can move up the scale as they get older. But it does give us a framework on which we can design treatments, so the patients with XLT have a much less likelihood of developing major complications, and therefore our treatments need to be less likely can be less invasive, whereas patients with Wiskott-Aldrich syndrome require definitive therapy, we believe, early on in life. And that is in the form of allogeneic stem cell transplantation, which Lisa is going to talk about in a lot more detail. So I'll skip that slide. So what causes this disease? Well, the gene encodes a protein which is expressed only in hematopoietic cells. It's called the Wiskott-Aldrich syndrome protein. It's a multi-domain protein which interacts with lots of other binding partners within the cell. And it's the founding member of a whole lot of other proteins that are responsible for organizing the actin cytoskeleton. So this is the structure, or one of the key features of the cell that holds it together and enables it to perform purposeful and efficient activities such as migration or cell signaling. Now, Wiskott-Aldrich syndrome protein is only expressed in blood cells, and that's why the phenotype of this disease is restricted to the hematopoietic system and the immune system, whereas the other proteins in this family are expressed more widely, and to date we are not aware of any mutations in these proteins that cause human disease, although of course it's possible. So how does this thing work? Well, the main thing that the Wiskott-Aldrich syndrome protein does is facilitate the polymerization of actin within the cell at appropriate locations. And the way it does this is by being activated by a whole lot of different signals, including from RAID GTPases within the cell, by post-translational modifications such as phosphorylation, and binding by from other partners. So whilst it sits in the cell in an off configuration or an auto-inhibited configuration by folding over its active domain at the end, which I can you see that? Yeah. So this terminal domain here, which we call the VCA domain, binds a complex called the ARP2/3 complex, which polymerizes actin. When WASP is resting in the cell and not doing anything, it folds in on itself and is inactive. When it binds activating molecules, that end of the molecule opens out and is then able to bind the ARP2/3 complex, and actin polymerization occurs. Now, one of the key activators is this tyrosine. It's difficult for me to see this actually. This tyrosine 291, which sits in the middle of the molecule. So when this is phosphorylated, the auto-inhibited domain opens out, polymerization occurs. And I just want you to remember that, because I'm going to touch on a totally different disease a bit later on, which is caused by mutations in this region of the molecule. This is the gene, this is the Wiskott-Aldrich syndrome gene, and really what I put this up to do is to illustrate to you that the mutations that cause Wiskott-Aldrich syndrome are distributed along the length of the gene. And it depends how destructive these mutations are as to the severity of the disease. So what we find is the mildest form of Wiskott-Aldrich syndrome, i.e. XLT, tends to be caused by missense mutations at the N-terminus of the gene, whereas the classical form of the disease tends to be due to nonsense mutations or stops variously spaced throughout the gene. Now, there's an interesting bunch of mutations. I keep losing this arrow. I can't see it. Oh, there it is. Right in the middle of the molecule here, which I've just alluded to, and these cause a different disease called X-linked neutropenia, and if I have time, I'll show you why that's the case. So what does WASP do in cells? Well, and this is important because we still, to this day, really don't understand why deficiency of the actin cytoskeleton causes immunodeficiency. We certainly don't understand really why it causes defects of platelet formation and increased platelet consumption. We know the actin cytoskeleton is important for cell shape, and cells are not static blobs within our blood. They are moving all the time. They require purposeful actin polymerization at localized sites to achieve cell migration, for example. In addition to that, we know that actin polymerization is important for assembling efficient signaling complexes so that cells can respond to appropriate extracellular stimuli and also intercellular stimuli. Now, one example of that is shown here. So these are dendritic cells that are derived in vitro from patients and from normal individuals. And the key thing I want you to note here in the top of the image is that these dendritic cells at the front of the cell, so this is the front that is moving forward on the test tube or on the glass slide, if you like, have these adhesion structures called podosomes. They're little feet that allow the cell to adhere efficiently to the substratum. Now, one of the notable things that we found many years ago was that when we derive cells from patients with Wiskott-Aldrich syndrome, they are completely lacking these structures. Now, these structures have integrins and other molecules in them, and so we felt on the basis of that it was likely that the cells or the immune cells or hemopoietic cells from patients are unlikely to be able to move normally and therefore respond to physiological stimuli. And I'll just illustrate that with this video if I can. So these are dendritic cells which we've just plated on a glass slide, and we've asked them to just move around and then put a camera on top to film them. And what you can see here is that these normal cells have a nice front, so that's the front where you would see the big podosome assembly of these attachment structures, and a tail which retracts to follow the front. And in doing so, these cells are able to move purposefully on the glass slide. Now, if you ask cells from a Wiskott-Aldrich syndrome patient to do that, you can immediately see there are differences. So they don't really quite know which is the front and the back. So if you look at some of the cells, like this one down here, it tends not to polarize normally. It doesn't have that nice leading edge of podosomes, which we know are absent in this condition, and it tends to elongate in the two directions. So if you actually map that out, you find that over a period of time, these cells don't achieve very good net migration. And on the basis of that, we, over 10 years ago now, felt that many aspects of this condition could be explained by deficiencies of cell migration, not just by dendritic cells, but also by other immunological cells like B cells and T cells. So therefore the concept arose that Wiskott-Aldrich syndrome may in fact be a cell trafficking disorder. Now, an example of that, which is clinically relevant, is shown here. So this is a study in mice, which we know is recapitulated in patients. And the key observation is that when we look in the spleens of these mice, so this is a normal spleen, and this is a spleen from a Wiskott-Aldrich syndrome mice, they're completely deficient in this zone of B cells called the marginal zone of B cells. So you can see the normal here, and the red area outside this follicle here is completely missing. And the reason for that is because we believe is because these marginal zone cells aren't able to traffic and adhere into the correct site within the spleen. So is that clinically significant? Well, it might be, because we've known for many years that Wiskott-Aldrich syndrome patients respond very poorly to polysaccharide antigens, and pneumococcal antigens, for example. We believe now that, in fact, the lack of the marginal zone is entirely responsible for this. So when we go into the mice, as well as humans, and try and vaccinate them with polysaccharide antigens, they just don't respond as well. Now, one question that we didn't know at the time was whether this is an intrinsic problem of the disease, or whether, in fact, that was secondary to other cellular abnormalities. So I think I'm not going to show you the numerous publications that have come out over the last 10 years analysing in detail the way that cells traffic between skin, for example, to draining lymph nodes, or between bone marrow and thymus. But there is very good evidence, I think, that in every one of these compartments, that in the absence of the Wiskott-Aldrich syndrome protein, that the trafficking is abnormal. And therefore it is likely that at least a contribution of the immunodeficiency of this disease is due to that trafficking abnormality. Now, I have one slide on thrombocytopenia, and we can talk about this later, because it is very difficult to explain the thrombocytopenia of Wiskott-Aldrich syndrome as a cell trafficking abnormality. And in fact, we really don't know what it is about a deficiency of WASP that causes the thrombocytopenia, apart from the fact we know that if you ask WASP-deficient megakaryocytes to make platelets in vitro, they seem to do it reasonably well. If you look in a mouse, in vivo, they seem to have some delocalization of platelet formation, which may suggest that there is some abnormal cytoarchitecture, and we do know that by looking at the cells in vitro. So, for example, megakaryocytes from WASP-deficient patients or mice don't make podozytes, so they don't adhere normally. The important thing about thrombocytopenia in this disease is that if you perform a splenectomy in patients, you can pretty much fix the problem. They will still have some, in some cases, numerical deficiency and structural deficiency, but to a large extent the thrombocytopenia is resolved by splenectomy. Now, I've said that the trafficking may explain some of the deficiencies in this disease. That's part of the story, but I think it's much more complicated than that, and we're just now trying to understand why these patients are truly immunodeficient, because that may help us design better therapies. So I'm just going to show you an example here. So these are Wiskott deficient mice which are challenged by LCMV, and the reason we do this experiment is because we know that patients with classical Wiskott-Aldrich syndrome manage herpes viruses pretty badly. Now, the result of this experiment is that after challenge, the viruses persist a lot longer, and there is a lot more cytopathic damage, which in this model is not caused by the virus itself. It's caused by infiltration of CD8 positive T cells. So the data in this slide really shows that the viruses, so this is measuring viral clearance, that the viruses in WASP deficiency persist a lot longer, and so the inflammatory process persists a lot longer. Now, why don't these mice clear the virus? Well, to cut a long story short, it's because they don't mount a normal CD8 T cell response. And that's measured in this slide here, looking at total T cells. It's looking at specific T cells using tetramers that will recognize T cells against the major epitope of LCMV, and also looking at interferon gamma positive specific T cells, again against major epitope recognizing cells. So why is this? Well, again, to cut a few experiments out of this and cut to the end important point, we know in this model that one of the key initiators of CD8 T cell immunity that is responsible not only for priming the CD8 cells but for maintaining a CD8 T cell response is production of type 1 interferons by CD11C positive dendritic cells. So when we ask Wiskott deficient dendritic cells to make type 1 interferons, we find they're not very good at it, both in vitro, and when we challenge mice in vivo and ask them to make type 1 interferons, we find also that they're very poor at doing this. So on the basis of this, I think we can be reasonably confident that one of the deficiencies in the T cell response in this disease is caused by a deficiency of dendritic cells, and we know it's restricted to that population to make sufficient levels of type 1 interferon. Now, we can give patients type 1 interferons, so it may be that that is a viable therapeutic option for future studies. Okay, let me just talk about B cells, because B cells are, we believe, to be important in the pathophysiology of this condition. I've showed you already that the marginal zone is depleted in this disease, and that is likely why these patients don't respond very well to polysaccharide antigens. So the experimental approach that we've used, and we're extending this to other cell populations, is to just to knock out WASP protein expression in B cells, and therefore ask what are the intrinsic deficiencies that are caused by that, and does that have any role in disease pathogenesis. So this is the experiment. We knock it out in B cells. We can look at then at B lymphoid structures within the mice. So maybe not unexpectedly, we find the marginal zone in these mice is completely depleted. So it looks exactly like the knockout mice, which says that this is an intrinsic B cell deficiency. But more interestingly, perhaps, is that when we look, I'll skip that one in the interest of time, but when we look at the production of autoantibodies in these mice, we find that, in fact, it looks exactly the same as knockout mice. And what I should have said at the start is that the knockout mice have very increased levels of autoantibodies. So this is an experiment looking at a whole panel of different autoantibodies. Red means there's lots of autoantibodies. Green means there's not very many autoantibodies. So on the left side of this picture here, let me find the arrow. There it is. This is a normal mouse. This is a mouse with a total knockout, all lineages, and this is the B cell knockout mouse. So you can see they're very, very similar in terms of production of autoantibodies. So this tells us, I think, to a certain extent, that B cells are important for generation of autoimmunity, both in mice and possibly in humans. And of course that's important because there are strategies to deplete B cells in humans that may be effective for treating autoimmunity. Okay, now very quickly, I'm just going to tell you about this other disease called X-linked neutropenia, because it's interesting. So this disease is caused by mutations in the Wiskott-Aldrich syndrome gene, but in a very specific part of it. And it's in a part that is responsible for maintaining auto-inhibition. So remember I said at the beginning that auto-inhibition means that WASP isn't active, it's not polymerizing actin. If you disrupt auto-inhibition physiologically, it can polymerize actin. And what these mutations do is actually disrupt this auto-inhibitory structure here and therefore make the protein constitutively active. Now, why does that cause neutropenia? Well, we know if we ask these proteins to polymerize actin in vitro, that they're much, much more active than the normal protein. So we know, at least in a test tube, that there's a lot more actin produced, and therefore, potentially, that could interrupt cell function. When we take cells from these patients, so bone marrow cells or grow colonies from bone marrow cells, we find that they don't grow at all. So somehow we've got to try and figure out how increased actin within cells causes this deficiency of myeloid cell proliferation. Now, it's not only a deficiency of myeloid cell proliferation, because we know that these patients not only are neutropenic, but have a susceptibility to genomic instability. And indeed, if you look at some of the very rare kindreds with this disorder, you find that several of them go on to develop acute leukemias. And when we look at these cells in vitro, we find that the cells have extra chromosomes, they're aneuploid, and some of them are even polyploid. So there's something that we believe about the extra actin polymerization is causing defects in mitosis. Now you remember that when cells divide, they constrict in the middle using a band of actin. And in yeast, that is a WASP-dependent process. Although in eukaryotic, in human cells at least, or mammalian cells, that's not the case. But still, the same process is responsible for splitting the cell in two and therefore segregating normal numbers of chromosomes. So we thought that maybe because of the extra actin within the cell, that this is going to be disrupted. And I just want you to look at this cell here. So this is a cell that is dividing. So this is a cell in vitro. It's a U937 myeloid cell. And you can see it divides in the middle, constricting actin band, and then splits in two, hopefully with a normal set of chromosomes. Now, if the chromosomes don't split normally, that cell will die because you activate checkpoints like the spindle assembly checkpoint. Now, what happens when we express the mutant protein in cells? So these are the same cell types, but now express the mutant protein. So increased actin in the cell, cells don't divide properly, and we will know that if chromosomes are replicating at the same time, the cells are going to become polyploid, and of course that's a great encouragement to the cell to activate apoptotic processes. Now, there are lots of reasons why that might be occurring, but our current belief is that this is a structural problem. So these are some pictures of cells which are clearly dividing. So you can see the blue here are the chromosomes, and the green are the microtubules just pulling the chromosomes apart during normal mitosis. So normally, in a dividing cell, from arrow, yeah, there it is, the actin is excluded from the nuclear region, and so you can see here it's just cortical around the cell edge. Whereas in cells that are expressing the mutant WAS protein, you can see it's stuffed throughout the cell, and in fact, is even between the microtubules where the chromosomes are separating. Now, we know that that's important from experiments I won't show you, because we know that in fact slows down chromosomal separation and also causes mistakes of chromosomal separation here. So what you can see here are some shadows of chromosomes. So this is a chromosome that's got stuck in the actin meshwork while the chromosome's been pulled apart. And and here's another one at the bottom, which is just stuck where the cell is trying to complete cytokinesis and split in two. And we know also from experiments not done by ourselves but by many other groups that this is a key trigger to cell apoptosis, to prevent aneuploidy. So I think on the basis of that we have a good idea that the cytoskeletal disturbances in the Wiskott-Aldrich syndrome contribute to the immunophenotype by multiple levels. So a certain component is due to cell trafficking disorders in different cell lineages. But not only that, there are clearly localization and signaling defects that contribute importantly, and they may be addressable by different therapies. And the second part, I showed you that dysregulated activation of the WAS protein results in cytopenia associated with increased levels of apoptosis. This is so-called X-linked neutropenia. So it's interesting. You have mutations in the same gene, one that turns it off, one that turns it on. You get a completely different disease phenotype. Now, what we don't know is exactly how this happens.
Thank you very much, Adrian.